All poisons are trendy. Part 2: Moleculogenetics and Immunohistochemistry. Neurotoxicity. As a neurotoxin, it affects the nervous system. Neurological function depends on depolarization of nerve and muscle fibers due to increased sodium-ion permeability of the excitable cell membrane. Lipid-soluble toxins such as beta-carotoxin act directly on sodium-ion channels involved in action potential generation and by modifying both their iron selectivity and voltage sensitivity. Beta-carotoxin irreversibly binds to the Na plus channels, which causes a conformational change in the channels that forces the sodium channels to remain open. Beta-carotoxin not keeps voltage-gated sodium channels open, but also reduces single-channel conductance. In other words, the toxin binds to the sodium channel and keeps the membrane permeable to sodium ions in an all-or-none manner. This has a direct effect on the peripheral nervous system. Batrotoxin in the PNS produces increased permeability, selective and irreversible, of the resting cell membrane to sodium ions without changing potassium or calcium concentration. This influx of sodium depolarizes the formerly polarized cell membrane. Beta-carotoxin also alters the ion selectivity of the ion channel by increasing the permeability of the channel toward larger cations. Voltage-sensitive sodium channels become persistently active at the resting membrane potential. Beta-carotoxin kills by permanently blocking nerve signal transmission to the muscles. Beta-carotoxin binds to and irreversibly opens the sodium channels of nerve cells and prevents them from closing. The neuron can no longer send signals, and this results in paralysis. Although generally classified as a neuron toxin, toxin has marked effects on heart muscles. These effects are similar to the cardiotoxic effects of the digoxin, a poison found in the foxglove plant. Toxin interferes with heart conduction, causing arrhythmia, extrasystole, ventricular fibrillation, and other changes which lead to cardiac arrest. Toxin induces a massive release of acetylcholine in nerves and muscles and destruction of synaptic vesicles. As well, petrotoxin R is more toxic than related petrotoxin A. Structural changes in nerves and muscles are the massive influx of sodium ions, which produces osmotic agents. It has been suggested that there may also be an effect on the nervous system, although it is not currently known what such an effect may be. Toxin activity is temperature dependent, with a maximum activity at 37 99 Fahrenheit. Its activity is also more rapid at an alkaline pH, which suggests that the unprotonated form may be more active. Given that this information is clearly that the toxin activity is at maximum rate at human regular body temperature, which is 37 degrees. So that is the reason why the V has to be stored below extremely low temperature because they try to inactivate the toxin or the virus, and then once it is injected into a human body, then the toxin or virus will be at its 100% activation. So, plus, I've noticed that in recent years, the mainstream trend is consume alkaline water, and they always promote that alkaline water is the best option for human body. But coming from the information here, they try to generate an environment within the human body to accommodate this toxin or virus, and so that the best environment for this to achieve the maximum activity. So, as a result, remember, don't follow the trend, don't listen to any mainstream trends. Now you have it. Alkaline water they promote within these past few years is for this very reason. Right now, for people who has received the inoculation and to be poisoned by the V. Toxin is an extremely potent cardio and neurotoxic steroidal alkaloid found in certain species of beetles, birds, and frogs. And this is from the Greek word fork. Structurally related chemical compounds are often referred to collectively as batrachotoxins. It is an extremely poisonous alkaloid. In certain frogs, this alkaloid is present mostly on the skin. Such frogs are used for poisoning darts. Batrachotoxin binds to and irreversibly opens the sodium channels of nerve cells and prevents them from closing, resulting in paralysis and death. No antidote is known. So now we know why the ancient cultures worship beetles and frogs. Issues: physical limitation of the ferritin. One of the main issues in Manito genetics is related to the physical properties of the ferritin. The ferritin is composed of 24 subunits of protein complex and a small iron oxide ore. The core of the ferritin is in the form of ferric hydroxide, which has antiferromagnetic properties. Some researchers have reported that ferritin has remnant magnetization due to their intrinsic defect and impurities. However, even with optimistic calculations, the magnetic interaction energy for heat or force generation is several orders below than thermal fluctuation energy. Recently, other researchers hypothesized that the iron oxide magnetic nanoparticles can generate sufficient heat or force with appropriate nanoengineering, such as tuning decomposites, doping atoms, or structures. Another attempt involves using higher hierarchical structure of nanocomposites. Researchers assembled the nanocomposites such as iron oxide nanoparticles or iron storage proteins to make the architectures with higher magnetic moment. Application: the field of magnetogenetics is a relatively new area of research that has the possibility to further the scientific understanding of how specific cells and genes contribute to the function of biological tissues, such as neural circuits in vivo, which is within life. It also has great potential for clinical applications as many medical tools that utilize magnetic fields, such as MRI, TMS, are already in use in patients. Remote control of gene transcription. Magnetogenetic techniques involve TRPV class receptors, which are selective calcium transporters, with magnetic nanoparticles or paramagnetic proteins, typically ferritin. The use of magnetic nanoparticles or paramagnetic proteins heated by a alternating magnetic field can stimulate the temperature-sensitive ion channel. When the local temperature rises, TRPV1 gates calcium 2 plus to initiate the synthesis and release of bioengineered insulin driven by a calcium 2 plus sensitive promoter. This method for remote regulation of gene transcription can be applied to basic research and potential therapeutic approaches for gene therapy, which is the current field. Wireless control of neuronal activity. The currently available magnetogenetics techniques combine targeted magnetic particle delivery with magnetic stimulation. This allows for the accurate spatial control of the targeted intervention. Inhibition or excitation of the target neurons in target brain regions. The temporal precision seems to vary depending on the threshold of ion channels and their frequency and the intensity of the stimulation experiments can be devised where the magnetic stimulation triggers some sort of cellular in vitro or behavioral in vivo. Viral vector is the most effective means of gene transfer to modify specific cell type or tissue and can be manipulated to express therapeutic genes. Several virus types are currently being investigated for use to deliver genes to cells to provide either transient or permanent transgene expression, which is the current V. Motor cortex, little genetics, freely moving mammalian behavior, and immunohistochemistry have been integrated to investigate the efficiency and validity of magnetogenetics as a neuroscientific tool. Magnetogenetic activation of the mice motor cortex has been shown to be effective in inducing locomotor changes. In freely moving mice, the torque-mediated magnetogenetics is a new toolkit established by Lee and colleagues that can transduce magnetic stimulation into mechanical torque for untethered neuromodulation at a long distance. In this study, scientists incorporated piezo-1, a mechanosensitive cut ion channel. To endow mechanosensitivity to neurons, piezo-1 is genetically expressed in the motor cortex via viral injection. After the expression of this ion channel, magnetic nanocomposites are injected into the same location. The nanoparticles generate and deliver the torque to the piezo-1 ion channel to stimulate specific neurons. The electrical signals are propagated through the neural network and change the behavior of live animal. That's why right now they're forcing us to take the V so that they can remotely control our behaviors, especially the upcoming zombie apocalyptic. What is fluorescent intensity? Fluorescence is based on photoluminescence, a process of glow, and light emission. It is a physical process in which light is emitted after it has been absorbed by a substance. The fluorescent intensity indicates how much light photons is emitted. It is the extent of emission and it depends on the concentration of the excited fluorophore. Fluorescence is created by the absorption of energy light by fluorescent molecules called fluorophores. The absorbed energy rises electrons to a higher energy level. As this excited state is unstable, the electrons fall back to ground state and thereby emit light. DNA and RNA qualifications, fluorescent intensity-based assays that quantify nucleic acids are very popular for sequencing applications. These require exact sample qualification with high specificity, as explained in our blog Next Generation Sequencing, why quantifying exact DNA RNA is important. DNA and RNA qualification assays are provided for different concentration ranges with different specificities, with different fluorescent properties, and by many life science chemistry providers. A comparison of different kits and how they perform on BMG lab tech readers is found in the application note. DNA qualification using absorbance and fluorescent methods. Can you see fluorescent with your eye alone? The most striking example of fluorescent occurs when the absorbed radiation is in the ultraviolet region of the spectrum and thus invisible to the human eye while the emitted light is in the visible region, which gives the fluorescent substance a distinct color that can be seen only when exposed to UV light. The green fluorescent protein is often used in genetics as a marker. Many substances, such as proteins, have significant light absorption bands in the ultraviolet that are of interest in biochemistry and related fields. UV-capable spectrophotometers are common in such laboratories. The evolution of early reproductive proteins and enzymes is attributed in modern models of evolutionary theory to ultraviolet radiation. UVB causes thymine base pairs next to each other in genetic sequences to bond together into thymine dimers, a disruption in the strand that reproductive enzymes cannot copy. This leads to frame shifting during genetic replication and protein synthesis, usually killing the cell. Here's something fun to read. Herpetology. Reptiles need UVB for biosynthesis of vitamin D and other metabolic processes, specifically cholecalciferol vitamin D3, which is needed for basic cellular and neural functioning as well as the utilization of calcium for bone and egg production. The UVA wavelength is also visible to many reptiles and might play a significant role in their ability to survive in the wild as well as in visual communication between individuals. Therefore, in a typical reptile enclosure, a fluorescent UVA-B source at the proper strength spectrum for the species must be available for many captive species to survive. Simple supplementation with cholecalciferol vitamin D3 will not be enough as there's a complete biosynthetic pathway that is leapfrogged, risks of possible overdoses.